Welcome to our lecture on Protochordata characteristics. Imagine you are looking at the blueprints for a highly advanced sports car, but instead of the final product, you are looking at the very first basic prototype. In the animal kingdom, you and I are the advanced sports cars, complete with complex backbones and skulls. Protochordates are those early, fascinating prototypes of our evolutionary lineage. Today, we will explore these simple, ocean-dwelling creatures and uncover the foundational blueprints that eventually led to all vertebrates, including humans. Now that we have introduced our topic, let us understand what exactly a protochordate is. In biological terms, Protochordata is an informal, paraphyletic grouping of marine animals. They are incredibly special because they exhibit the fundamental characteristics of the phylum Chordata, yet they completely lack a true backbone or a skull. You might wonder why we spend time studying tiny, brainless sea creatures that live buried in the sand. Their importance lies in their role as the ultimate evolutionary missing link. They provide living proof of how complex vertebrate systems evolved from very simple structures. Building on their role as an evolutionary bridge, let us look at the specific features that define them. To be classified within this group, an organism must display 4 hallmark traits at some stage in its life cycle. First is a flexible supporting rod called a notochord, which provides structural support. Second is a dorsal hollow nerve cord, the evolutionary precursor to our spinal cord. Third, they possess pharyngeal gill slits, which they use primarily to strain microscopic food from the water. Finally, they have a postanal tail. Every single vertebrate, including you, had these 4 traits during early embryonic development. Having listed the 4 hallmark traits, let us now see exactly where they are located in the body. On your screen is a schematic diagram of a generic protochordate. Notice the long blue tube running along the top? That is the dorsal hollow nerve cord. Just below it is the notochord, acting as a flexible skeleton. In the throat region, you can clearly see the pharyngeal slits, which act like a sieve for filter feeding. Finally, at the far right, the body extends into a postanal tail. This simple yet highly effective body plan has allowed these creatures to survive for hundreds of millions of years. Following our look at their anatomy, let us explore how these animals are categorized. The Protochordate group traditionally includes 3 subphyla: Hemichordata, Urochordata, and Cephalochordata. Unlike true vertebrates that developed complex skeletons and jaws, these animals retain a relatively simple body plan. Because they lack a bony skeleton, their structural support comes entirely from the notochord, or in groups like the Urochordates, a tough outer covering called a tunic. This simple classification helps biologists group them based on which chordate traits they retain into adulthood. This simple body plan leads us to a fascinating realization about our own bodies. The importance of protochordates lies in their role as the ultimate evolutionary missing link. They provide living proof of how complex vertebrate systems evolved. For instance, the complex human spinal cord directly evolved from their simple dorsal nerve cord. Similarly, the human thyroid gland evolved from a simple feeding structure called the endostyle found in these creatures. By studying the genetics of these organisms, scientists can deeply understand human developmental biology and even the origins of congenital birth defects. Beyond their evolutionary ties to humans, these animals play a crucial role in their own habitats. Since protochordates lack complex jaws and limbs, they survive primarily through continuous filter feeding. They use their pharyngeal slits to constantly strain microscopic food particles from the surrounding ocean water. Because they are highly sensitive filter feeders, they serve as excellent ecological indicators for the overall health of marine environments. By monitoring their populations, researchers can effectively track ocean pollution and measure microplastic accumulation in our fragile marine ecosystems. Understanding their ecological role brings us to how they actually feed. Since these animals rely entirely on filter feeding, let us look at the mathematics behind their survival. We can deeply understand their feeding efficiency by calculating the volumetric flow rate of water passing through their simple bodies. The formula is Q equals A times V. Here, Q represents the volumetric flow rate of water. The symbol A stands for the cross-sectional area of the animal's mouth or siphon. Finally, V represents the velocity of the water being drawn into the body. This formula tells us that the total volume of water an animal can filter depends directly on how wide its mouth is and how fast it can pump the water. If the area increases, or if the pumping velocity increases, the total amount of food-rich water processed will also increase. Before we move on, let us see this formula in action with a concrete example. A marine biologist is studying an adult sea squirt attached to a coral reef. We are given that the cross-sectional area of its siphon, A, is 2.0 square centimeters. The velocity of the water being pumped inward, V, is 3.5 centimeters per second. We need to find the total volume of water filtered every second, which is Q. We choose our flow rate formula because it perfectly relates physical size to water speed. We substitute 2.0 for area and 3.5 for velocity. Multiplying these together gives us our final answer. This means that every single second, this simple protochordate is actively filtering 7 cubic centimeters of ocean water to extract tiny plankton for its survival. Now that we have used the standard formula to find the flow rate, what happens if we already know the required flow rate, but need to find the water speed? We must rearrange our equation. We start with our core formula, Q equals A times V. To isolate the velocity variable, we divide both sides of the equation by the cross-sectional area, A. On the right side, the area terms cancel out perfectly. This leaves us with a new, rearranged formula, V equals Q divided by A. This mathematical rearrangement is crucial because it allows us to work backward from an animal's nutritional needs to determine exactly how hard its cilia must work to pull water inward. With our newly derived formula, let us solve a problem involving a lancelet buried in the coastal sand. To meet its daily nutritional requirements, this lancelet must achieve a flow rate, Q, of 12.0 cubic centimeters per second. Its mouth area, A, is very small, just 1.5 square centimeters. We need to calculate the required pumping velocity. We use our rearranged formula, V equals Q divided by A, because we are solving for speed. We substitute 12.0 for Q and 1.5 for A. Dividing 12 by 1.5 gives us 8.0. This indicates that the lancelet must maintain a continuous water current of 8 centimeters per second to avoid starvation. We have covered a lot of ground today, so let us review the most important ideas. First, remember that protochordates are the crucial evolutionary stepping stones between simple invertebrates and complex vertebrates. Second, even without a true backbone, they proudly display the 4 defining chordate traits: a notochord, a dorsal hollow nerve cord, pharyngeal slits, and a postanal tail. Finally, we saw how their survival relies entirely on filter feeding and how simple fluid mathematics dictates their biological success. By mastering these characteristics, you now hold the key to understanding the very origins of your own vertebrate lineage. Great job following along, and keep exploring the wonders of zoology.